All right, so in this one we're going to talk about endo and exocytosis. So endocytosis, endo means in, cyte means cell, and osis is process of. So it's the process of bringing something into the cell, some kind of yummy that the cell wants, maybe some food, maybe some hormone that it needs or whatever. And so it is going to, as the cell approaches it, the cell's going to start to extend its cell membrane around that. It's going to close around that, so it's going to pull those membranes around so that now your yummy, whatever it is in a vesicle, and then that vesicle is going to break free of the cell membrane and head into the cell to do whatever. And I say this is some kind of yummy. This is also how your macrophages get rid of bacteria. So this could be some kind of yucky. This could be a bacteria, and this is one of your immune cells that's going to gobble it up and kill it. There are 3 types. This one that I just showed you about is phagocytosis, which is just the eating of food, so some kind of solid. This is supposed to be a drop of water. Y'all know my art skills are not great. This is called penocytosis, and that is when you're bringing in a fluid. And if you're bringing in water, it's rarely that you actually need the water or that the cell needs the water. It's that it needs what's in the water. So it needs something dissolved, some solute in the water. And then the 3rd type is receptor-mediated endocytosis. And this is where you need some kind of whatever, it binds to a receptor. And then after that happens, whoopsie, I didn't make my membrane very pretty, that starts to form a vesicle and it will close, but you've got to activate that receptor for that to get started. So that is, I can explain the process of endocytosis, including the structures involved. That would be the cell membrane and a vesicle. And I can discuss the 3 main types: phagocytosis, penocytosis, and receptor-mediated. All right, I can explain the process of exocytosis and the structures involved. So exocytosis is the process of bringing something out of the cell. So this is some kind of waste product, or maybe it's a hormone or a protein that needs to be released. The Golgi has packaged it up in a vesicle, so those are 2 players involved. That is going to get shipped to the cell membrane. It is going to bind to the cell membrane. It is going to then open up, fuse with the cell membrane, opens up, and releases those contents out into the extracellular space. And so that is exocytosis. When this happens, though, let's say that you had, let's do another one. Let's say that you had a receptor that you wanted to put on the cell membrane's surface. I don't know if you can see that. So it was pointing on the inside of the Golgi. And then when the Golgi made a vesicle, it was pointing on the inside of that vesicle. And then when that vesicle fused with the cell membrane, now, let's bring that over here. So here's that vesicle. Here's that receptor. Whoops, drew it backwards. There's that receptor. It kind of looks like a letter Y. Cell membrane is going to fuse with the cell membrane. And as it opens up, now that receptor that was on the inside of the vesicle is going to be on the outside of the cell membrane. So this is in, this is out. Okay, in, out. And so it says, I can describe the process involved in moving a protein from the ER to the Golgi and out of the cell. So it's going to be made by the ER, packaged up in the Golgi, and then sent through exocytosis in a vesicle to the cell membrane. And it will, if it's on the inside of the vesicle, or if it's inside the vesicle, it'll be released out. Or if it was outside of the vesicle, it would be, and that would only be a receptor, it would stay on the inside. So we're seeing diffusion or movement of the vesicle, which is not diffusion. I shouldn't say that, because it does require that motor protein to walk the vesicle to the membrane, and then exocytosis for it to actually escape out of the cell. And so those are the processes and structures involved in exocytosis.